Okay, thank you very much. It's my pleasure to be here. For me, the last visit Bangkok is 2020 February, so 2.5 years. So very happy to be here. Okay, so today's my title is Innovation in Cell Manufacturing for the Regenerative Medicine. So mainly for the machinery system to produce the cell I want to introduce. But before starting today's topic, I want to explain what is the regenerative medicine in Japan. So 8 years ago, we have a new regulation for the cell therapy. So there are two types of the cell therapy. One is a conventional system supported by the public healthcare insurance. But another one is medical practice using the processed cells. Okay, loudly. Okay. So in Japan, the approval products already 14 products now. So 12 is autologous transplantation system, and 2 is the allogenic transplantation system, such as the mesenchymal stem cells. So now we have 14 products at the moment. These products already covered by the public healthcare insurance in Japan. So after establishment of this two regulation in Japan, we start to consider who is the stakeholders, because the stakeholder is important to understand what is the cell therapy and the regenerative medicine. So in Japan, Japanese Society for Regenerative Medicine, I'm one of the director in this society. So we thinking about who is a stakeholder. Of course, patient and volunteer, and also the creators to prepare the new regenerative medicine. And also the conductors, this is medical doctors, and also the technical supporters, such as me, engineer. And also we often discuss with the regulator that these 6 stakeholders gather to start what is the cell therapy and what is the regenerative medicine. So by thinking about this one, we start to we prepare the many education system, not only for the medical doctor, but also the engineer or cell processing processor operators, and also the company person, and also the patient. And so this is the most important to improve the real world of the stem cell field. In Osaka, our laboratory starting the new project, I want to say the research base for cell manufacturability. So in this research base, we now 6 companies gather to start what is the research ecosystem. One of the company supplying the facility, another company prepares the automation system, and several companies is for the cell manufacturer. We gather to discuss. In this research base, we're thinking about the 3 points. One is, of course, developing the system developing for the manufacturing. We want to realize the automation system for cell processing for the clinical application. But for this purpose, we also need the regulation. So that's why I joined the Japanese government to establish the regulation. And also as an academic position in university, we start to consider what is the human resources development for the future researchers, such as a student, and also the company person to realize a new field. So we often say the kotozukuri in this case in Japanese. And also we collaborate with not only Osaka University, but also collaboration with other company for the preparation of the training system in Japan. And after that, we start to consider what is our mission in the laboratory. In our laboratory, my background is chemical engineering, not biologist. So my standpoint is how to prepare the cells for the patient. That is my main topic. So I often use stem cells such as iPS cells or mesenchymal stem cells, but my interest point is how to prepare the cells, especially for the culture system. So here is several video image, such as the automation system, culture system, and sometimes we need some computational system to predict when our product can be prepared. Before starting today's main topic, I want to insert several slides because our laboratory, not only for the development of the cell processing, but also considering what is the tissues we often consider. So in Japan, in Osaka University, Professor Sawa established a new system using the skeletal muscle cell sheet for the cardiac failure. So this technique already approved by the in Japan, Thermo company. And patient can use this system using the autologous skeletal muscle cells. So and now in Japan, from 2020, Professor Sawa started to prepare the cardiac muscle cells. They are derived from the iPS cells. In Japan, already starting the clinical research. And so that means not only skeletal muscle sheets, non-beating cells, secretion of some cytokines after transplantation, but also the beating cells for the clinical application. For this purpose, as Professor Kim, name is Kim? Okay. Kim already mentioned some responsive surface. I also collaborate with Professor Okano in Tokyo Women's Medical University using the thermoresponsive surface to prepare the cell sheet. So using the stackable sheet, we often use a 5-layer sheet, looks like the tissues. So we start to consider how to understand the phenomena in vivo. I mean that we try to prepare in vitro assay system. So we use skeletal muscle cells and prepare the 5-layer sheet and putting the Hubeck cells, endothelial cells, to make some network formation of the endothelial cells. Looks like the pre-vascularization. So using the optical technique, such as wider observation system, we understand how much the endothelial cells network obtained in the 5-layer skeletal muscle sheet. So from this technique, my former student, she's now here. So she prepares some quantitative method because we need the evaluation system, how long the network system occurs. Very surprisingly, if we add the FGF, basic FGF, that's very famous for the vascular formation. So compare with without FGF, in the sheet, they have endothelial cell connected each other. Longer connection occurs in the sheet. So, but we don't know the reason why. So that's why we try to have some dynamic imaging system like this. So the left side is upper side is a control, and the bottom side is addition of the basic FGF at 60 hours, at 86 hours. I mean, maybe you find many red stir and the white stirs. Bottom side is much less the white stir, because the white stirs is a disconnection position after connection. So that means the addition of the FGF causing the disconnection, prevention of the disconnection. So that means how to say this addition of the FGF promote the and keeping the endothelial network system in the skeletal muscle cell sheet, so longer time. So this is the first understanding and the role of the basic FGF in the tissues. So that normally if we didn't prepare the tissues, of course, the FGF we can add and understand it well. Most of the research are targeting for just a number of the endothelial cells. But in the sheet, practical sheet, if we prepare, we can understand the real phenomena that looks like the mimicry system of the in vivo system. So that's a kind of the one of the topic today. And from here, I want to move to the another topic, main topic. So I want to mention the one keyword. So one keyword is cell manufacturability. Maybe you don't know this term, but the manufacturability is well known for the engineer. So just how to prepare the product stably and efficiently. I mean, saving the cost and stably. So, but we thinking about the first amino cell manufacturing. So that's why we start to consider definition of the cell manufacturability. I define the capability of cell manufacturing through the process bridging between biological aspect and process aspect. Because as an engineer, we want to produce the cells. The production, we need the reproducibility and stability. That's our mission. So, but we have to understand the cell properties. We also often consider why the cell don't want to grow during the culture. Maybe some of the students and the researcher don't want to do the cell culture. I don't like the culture, honestly. I like cell, but I don't like because it's sometimes difficult to obtain the same result. That's my starting point to understand. And the design for the cell manufacturability is manufacturing design cell-based product in such a way they are easy to manufacture through the simple, safe, and efficient, I mean the cost-saving process with stable product quality and secure customer by considering transportation and preparation outside the factory. Okay, so we want to realize a good culture cell manufacturing based on this idea. Why we have to consider like this? So for the production, as a product output, we want to prepare a cell-based product for the clinical application, but the product always have a same quality. That's a very important mission. So to prepare the same product, we consider the variation, quality variation for cells, ancillary materials such as the medium and the vessels as an input. If the input start to fluctuate, easy to get the bad quality of the cells. So, and also from outside of the process, we often get some different variation of the temperature or aseptic environment. That means a kind of extrinsic noise. The extrinsic noise enter into the process will fail the product. So, and also that's a conventional consideration, but we also have to consider during the process, cell easy to make some fluctuation. So I often say the intrinsic disorder. So we don't know the cells. That for the production, very difficult to keep prepared, maintain the same quality. So these consideration is important to prepare the same quality of the product, cell product. Why we obtain the different quality? Actually, left side is our laboratory member, expert of the culture, and on right side the practical culture system in the cell process facility. So what is the motion during the culture, such as a medium change. So we need the aseptic technique and the motion stability as well as a prompt operation. Okay, so these are important to keep the same quality for the during the cell processing. So from here, I want to mention the practical machinery system. This is a kind of the isolator system. So isolator system is a have a robustness of the aseptic environment. So this here is a pass box. After introducing the materials, we can start to have some operation here, and with the incubator and the centrifuge here. This house system is well known in the world. So during the process, the manual operation very easy to fluctuate the operation. So that's why how to improve this system. So the right side, we developed a new system in my laboratory. So there are two systems, manual operation module and automated operation module with incubator. Between module, we can transfer the materials under the aseptic environment. So like this. So between modules, aseptically, we can bring the materials using this transfer module to connect each other to open under the aseptic environment. And if necessary, we can bring to the incubator to the other place to operate independently to supply gas and electricity and exchanging the signal. And if necessary, we can connect to the other modules freely. And also, of course, automated, modules, we can realize a robotic operation. Sorry. So we realize several we prepare the practical facility. Unfortunately, we don't have enough money to establish the facility. That's why just having several tools to realize this system. But you know what is the automation system? What is a manual? What is a machine operation? What is a manual operation? So we have to consider which is better. Maybe if you have some experiences for the cell culture, for example, thawing from the freezing place, that's a quick operation with several minutes, not the 1 hour level. So that we cannot use the automation system because the automation system has start to consider how to quick operation. So that's why we prepare the manual operation and the machinery operation. But maybe you don't like the culture, even though the Saturday and Sunday, sometimes you have to come to the laboratory for the medium change like this. So the automation system can possible, okay, automatically. That's a good feature. So tedious work, it's much better to ask to the machinery system, but quick motion, it's much better to ask the manual operation. So we distinguish between operations. After several development, we try to prepare the machinery system. But before establishment of the machinery system, first we have to realize the machinery system in computer, not the practical field, because how to prevent the hindrance and also how to move. So in computer, first we design the space for the manufacturing, and then practically we start to consider practical machine. But you know, what is a good motion? Sometimes beginner fail the culture, but I don't know the reason why. Okay, so that we want to measure the motion. Younger person having some game, TV game, such as a Wii or PS4 or something like this in Japan. Actually, my children often use TV game. From the TV game, recently shaking the remote controller, and in the TV, you find the same motion. So using this technique, we measure the motion during the operation. So we install the motion capture sensor in the dishes, and we compare the manual operation and the automation system and to improve the motion. So the right side is the manual operation, left side is the automation system. We measure the gravity force for XYZ axis using this motion calibrator, which can measure the motion. So unfortunately first time, left side, very high gravity force compared with the manual operation, but we know the way. So that we improve the motion for the higher value of the gravity force for the machinery system. And we try to improve the motion. Actually, the 4.4 is a very high roller coaster system. In Japan, 3.67 is the highest. Okay, very dangerous roller coaster. Some of the people start to have an injury recently. So the much higher operation in the automation system occurs. So so we try to improve the system, and finally, after improvement of the motion, we can realize the same as the manual operation. Okay, so that's why we prepare the good motion system using the robotic system. To apply the practical culture, we try to prepare the culture system for the retina epithelial pigment epithelial cells derived from the iPS cells. First, you have to see how long, 117 days. My student don't want to do. Okay, no holiday. Actually, every day have to change medium. So no student, just gathering experts to realize. During the process, especially for the differentiation into the retina pigment epithelial cells derived from the iPS cells, the very fragile structure exists. Layered structure because of the differentiation from the undifferentiated state to the neural stem cells and also the retina pigment epithelial cells. So the very fragile, the bad hand sometimes causing the destroy of this structure, causing the different fate in future. So we want to realize gentle motion using the robotic system. So fortunately, we can realize several automation machinery system from the after thawing. Thawing is a manual operation, but after thawing, medium change, passage, and filtration and dispensing, we can realize a machinery system like this. So how about the stability of the process? My expert is an expert, okay, but they unfortunately not student, expert. During the 6 rounds, only one time successful. Unfortunately, because normally expert prepares 6 wells, smaller size of the vessel. But for the practical production, we move to the just 100 millimeter in diameter dishes, not so big. But even though that this scale up, the experts start to fail. So that's a practical situation. But fortunately, machinery system, I don't know the reason why, but the first time successful. And of course, same motion. So that means we can realize successful performance during the process. But unfortunately, last time, I don't know the reason why. But compared with the manual operation, the robotic system is possible to make the stable process. And finally, we get the stability of the process, and we get retina pigment epithelial cells. That's a black eye part. We can harvest the cells successfully using the machinery system from after thawing to the end of the culture. Okay. So I want to close, but before closing, sorry, maybe I want to have some message. So the first we have to consider if we want to establish the culture process, we have to consider cell manufacturability because we need some aspects, not only biological aspects, but also the engineering aspect necessary to prepare the good performance. And also today I didn't mention the automation system is a very good system to reduce the cost. Because if we start the practical manufacturing, if we apply the expert, we need the backup of the expert, and the education fee is very high. So, but in case of a robotic system, once after installing, easy to maintain. So that's the last slide. So this work supported by the many Japanese government and also our laboratory member. Fortunately or unfortunately, we have more than 35 students now, and supported by the only 3 staffs. So staff always crying, and also other company person come together to become a very large group. Okay, thank you very much for your attention.
Very informative and wonderful presentations. Again, anyone here has a questions? Maybe I have a question. This is my curiosity. Like when you compare about machine manufacturing and manual, like humans manufacturing. So one the pain point of people who do a sales culture is the contaminations. What about the benefits of using the machines for cell manufacturing compared to the manuals? Yes.
Okay, thank you very much. Today I didn't mention, but if we prepare the first chamber isolator system, I often say this is a very high robustness for the prevention of the contamination, because that's a practical machine. I'm one of the member of the ISO for the aseptic processing. Actually, this isolation system documentation, I'm become the world project leader to establish documents. So compare with the other system, such as a biosafety cabinet, quite different. First, if you use a biosafety cabinet, please understand the flow and the robustness. I didn't deny the use of the safety cabinet. Our laboratory use many biosafety cabinet, but first you have to understand how to use. Okay. Actually, can I mention more? So because one student come to the operator, that's a bad situation for the contamination. Because in front of the safety cabinet, once after coming somebody, and the motion comes and the flow enter into the inside the safety cabinet. That's a safety cabinet. So that you have to keep alone if you start the operation. That's my message.
Thank you very much.